We're going to be looking at active transport today. Here's a couple definitions here for you. So when we're looking at movement of particles from areas of low concentration to areas of high concentration, we know that that is going to be active transport. This means that we are now going against the concentration gradient rather than with it, and therefore it does require energy. For active and passive transport, I really like to think about kids playing in a field, okay? So follow along with me here, right? So we've got these kids and they're playing tag in a field. Now, what if we put them in a really, really big field? If we have a huge group of kids just chilling in the middle of the field and tell them someone is it, they are going to run around just like particles in a solution would run around. They're going to start bouncing off of each other and filling up all corners. So those kids have energy, right? They're moving all around, but we're not using energy to dictate where they're going to go. They're going to end up going from areas of a lot of kids to where there's areas of fewer kids, right? And they're doing this from high concentration outward in all directions until they've basically filled up the field evenly. That is passive transport. So when we're starting to talk here about active transport, we're doing the opposite. We are rounding those kids up and putting them back all together. So we're taking these particles that are kind of randomly assembled and we're moving them against their concentration gradient. We are moving them to produce really a concentration gradient and rounding them all up, or in the case of membrane transport, getting them all into one area. And so we can take a look at a comparison here between passive and active transport. On the left are the types of passive transport that we have discussed so far. So we are looking at passive, and what's important to note about all of these diagrams is that we have an area of high concentration. In this case, it happens to be on top, but it doesn't matter where it is, so long as you understand that this is the area of high concentration and particles are moving to low concentration. We can look at simple diffusion, where we're moving the particles directly across the phospholipid bilayer, or some type of facilitated diffusion, in which case we're using some kind of channel protein that's going to allow very specific particles to move through. But in the case of active transport, things are a little bit different. In this case, what you're going to notice, getting a pen here, is that we've got a high concentration of these diamond shapes down here below, and we've got a high concentration of the circles up above. So the movement of these diamond-shaped things from where there's a low diamond concentration to now where there's a high diamond concentration, and of course we're also doing this with the circles. It just happens to be occurring in two directions. These are going from low circle concentration to high circle concentration. That requires energy. So that is really where we're at today. How do you establish a concentration gradient? You need to have energy. And in terms of the types, there are two types of active transport. When we look at active transport, we can be looking at pumps, or we can be looking at something called bulk transport. We'll start with the easiest, which are the pumps. Now, this is a nice simple diagram of a pump. And pumps are a form of molecular movement. We're just moving molecules or particles from one side of the membrane over here to the other side of the membrane over here. It's just this time we're going against the concentration gradient from where you have less, right? There's fewer of these guys here and more over here. So we're going from low to high concentration, again using energy, usually in the form of ATP. Why would you need that? Well, I don't expect you to really follow most of what's going on in this diagram, but what I do want you to see is that there is a difference in concentration, and this happens to be a neuron. Again, not so much for you to memorize, but a nice illustrative example here is that, remember those circles that we were talking about? There's a high concentration of circles up here, and there's a high concentration of diamonds down here. Well, as it turns out, in a neuron, that's really, really important. In this case, we're going to be looking at sodium and potassium. So we've got the sodium being represented by these little circly bits up here, and they are moving against their concentration gradient. They are moving from inside the neuron, where there is a low concentration already of them, and a greater concentration gradient is being established by pumping these little guys out through there. And then in the reverse direction, we've got potassium coming in, right? So we need this concentration gradient to be established so that the neuron can be ready to fire. If the neuron's not ready to fire, we, would die. So that's a big fail. And specifically that pump that is being shown in the previous diagram is something called the sodium-potassium pump. I don't need you to memorize the details. I do need you to just have an understanding here of why this works and why it's so important. So we've got our sodium-potassium pump. And it works kind of like any other pump by changing shape. And each time it changes shapes, it's going to allow the movement of whatever is inside of it. So if you have a sump pump in your basement, for example, right, here's a side view of your house. And here's your basement down here in the lower part. Now, if you have water that's coming in from an area of more water to less water, you're going to have a sump pump. And what that pump is going to do is allow the water to come in and then get pushed back up. Okay, so the water is going down and getting pushed back up against the force of gravity from where there's less water to where there's more. And of course, that is going to require this guy right here. When we're looking in the cells, we're using ATP. So in this case, we're going to look at 3 sodiums that are getting constantly taken from the inside of the cell and pumped out. In the reverse order, we're going to have 2 potassium that are on the outside that are then used to pump in. This is an illustrative example. It is not something that you need to memorize how many sodiums, how many potassium, how the whole process works. I just like that the diagram shows you that we have binding sites for those sodiums right there. Those get bound to it. ATP causes a change of shape that's going to release them on the opposite side. It just so happens that this is a 2-way pump. Again, details, nothing quite so important that you need to panic about, okay? So those are our type of pumps. Here we go. Pumps are anything that are going to be moving molecules from an area of low concentration to high across the membrane. But remember, there were another type of another type of active transport. In this case is bulk. And to understand bulk transport, we need to take a look at the endomembrane system. Yes, we have talked about it ad nauseum, and here we are again talking about it. The endomembrane system is going to consist of the endoplasmic reticulum, the Golgi, and all of the vesicles that are produced. Okay, this is really cool. We move a lot of things throughout our cells using the endomembrane system, and this is called vesicular transport because we're moving materials into vesicles. Here we go. So we've got the endoplasmic reticulum, this guy right here, okay? The membrane, this membrane can pinch off, and I'll show you, I'll post a video onto Schoology that you can take a look at. So those membranes pinch off, and that's going to make a vesicle. That vesicle separates away from the ER, and it's kind of carrying whatever, and then refuses over here with the Golgi. So essentially whatever was in the inside here, inside of that vesicle, now becomes part of the inside of the Golgi. And guess what? It's going to kind of move through each layer, just like we saw in the ER. It's getting pinched off and refused and pinched off and refused until finally everything on the inside has been appropriately modified, and what we need is now produced. And so then we can pinch off parts of the Golgi into a vesicle and then move it. Okay, so that is called vesicular transport. It happens within the cell, but it is also how we can move a great deal of material, a bulk amount, into or out of cells. So that is where we're going now. With this whole idea of bulk transport, it's all about taking vesicles and either merging them with the cell membrane, the plasma membrane, or taking the plasma membrane, letting that wrap around something in the environment and taking it in. So we can look here at bulk transport. And this is one type of bulk transport called endocytosis. That prefix there, endo meaning into, and cytosis meaning cell, and then the process of. So something that is bringing stuff into a cell. We can look at 3 types of endocytosis here. The first one, we're going to have the plasma membrane that kind of wraps up around some kind of large chunky substance, and much like eating, this is called phagocytosis, because that prefix there, phago, means to eat. So we're taking in materials. This is really important for your immune system. When you have bacteria, your white blood cells are going to phagocytize or engulf or wrap their membranes around bacteria and then nom it up and digest it. Okay, so phagocytosis is a type of endocytosis where we are bringing material into the cell. We can also look at cell drinking, which is called pinocytosis. In this case, a little vesicle starts to form and a high concentration of material from the environment is brought in. Now notice something really important here. We are not really bringing it into the cell's cytosol or the cytoplasm, not into the liquid itself. It's going into a vesicle. Now that can still allow it to potentially diffuse through the membrane or whatever, but it is not yet crossed a membrane if we've just done endocytosis. It has not done this. Ooh, some weird symbols going on, right? That's an arrow crossed out. We've not brought materials directly across the membrane. Instead, we've only put them into a vesicle. And then finally, on the far right, this is a specialized type of endocytosis that is going to use chemical receptors right here. So each one of these little Y-shaped guys is going to bind to a specific type of particle, and that's going to allow that particle to exist in really, really low concentrations outside of the cell. But each time one binds, that's going to kind of signal for the inside of the cell to start endocytosing it. That's going to pull it into a vesicle, and now we've got a really high concentration of that substance instead of a really low one. Okay, when we're looking at endocytosis, we are making vesicles, but we can also have a vesicle fuse with the outside plasma membrane, and that process right there is called exocytosis. Exocytosis is going to allow vesicles full of some kind of material to be brought to the surface of the cell. In this case, we're looking at a neuron, but it happens in all types of cells in the body. So we're going to have this vesicle here. It's going to fuse with the plasma membrane, and everything that was on the inside of the vesicle, all these little red dots, they then come out. Okay, so it's a great way to secrete something, and in fact, it's how your anterior pituitary and your hypothalamus and all of those endocrine organs that we looked at, your pancreas, that is how these organs are going to secrete their hormones into the blood vessels. So they're going to take a little pocket full of whatever the hormone is, go to the plasma membrane and merge so that they can pop it out. Okay. Boom. I don't know why there's a picture there. Anyway, so let's kind of recap what we've talked about so far. We've been looking at transport. Here's what I want to do, just like we do in class. As I'm going through these, if you need to pause, take a second and think about what I'm about to say, right? See if you can predict it. So when I'm thinking about transport, there are 2 types. See if you can list them and compare and contrast them. Okay, so we got 2 types, passive and active. The difference being that passive is going to have the movement of particles from an area of high concentration to low, down the concentration gradient, which does not require energy, down concentration gradient, while active transport is going to be the movement of particles from low to high concentration, which will establish a concentration gradient, and that therefore does need energy. Concentration, gradient. I'm sorry, I'm abbreviating so much because I want to look at the different types. So in your head again, go ahead and see if you can identify what the types of passive transport are that we have talked about in class. There are 3 of them. We looked at simple diffusion, which is the movement of particles directly across the phospholipid bilayer, and those are going to be things that are small and nonpolar. We looked at facilitated diffusion and osmosis. In both cases, we're going to need some kind of a channel protein to be able to allow molecules that are either too large or polar to be able to move through. And in the case of osmosis, that is specifically the movement of water from the area that is hypotonic to hypertonic. Okay, hopefully you got all of those, and we can go ahead and move on over and look at the types of active transport. Again, we're looking at things that move molecules from an area of low to high concentration, and that is going to require energy. So 2 types, list them out in your head and see if you can compare them. Okay, in the case of pumps, we're looking at anything where we have the cell membrane and we actually have molecules that are getting moved across them through some kind of pump, obviously. But in this case, we're going from an area of low to high. In the case of bulk, we are not moving across the membrane itself, but instead, wrapping the membrane around a vesicle. Okay, now when we look at bulk transport, again, 2 major types of bulk transport that we looked at. Anytime we are bringing material into the cell, that is endocytosis, right? We're bringing materials into the cell to create a vesicle, whereas in exocytosis, we are pushing things out of the cell by taking a vesicle and fusing it with the cell membrane. Finally, last one for you. Can you name the types of endocytosis that we looked at? Okay, phagocytosis, you should be thinking about cell eating. In other words, we're taking big chunks, for example, bacteria. And penocytosis is cell drinking, so we're taking materials that are insolute. And bonus points for you if you got that last one. Can you think about it? Yeah, I never explicitly said it, but that is receptor-mediated endocytosis. It was in the reading, but I wouldn't necessarily have you remember that one quite as well. But what I do expect, hopefully, you remember is the terms phagocytosis, penocytosis. Both of those are types of endocytosis, which means bringing materials into the cell. It is the opposite of exocytosis, but both of those are forms of bulk transport. Okay, that was the bell. There we go. And then I would expect you to remember that bulk transport and pumps are both forms of active transport, and active and passive are the 2 ways that materials can move into and out of cells. Okay, I'm trying to make this so you can hopefully see the whole thing on your screen. If you've got questions, email me, and hopefully this made sense.